Welcome to Mendel's Genetic Legacy, where we trace the foundational principles of inheritance and their modern applications. In this presentation, we'll connect Mendel's simple pea experiments to the big ideas that shape genetics, highlighting key experiments, laws, and the ways these discoveries still influence biology and medicine today. Mendel chose Possum sativum for good reasons. Peas have distinct, inheritable traits, short life cycles, large numbers of offspring, and can be easily self- or cross-pollinated with minimal environmental interference. Those practical advantages let him isolate single variables and apply statistical analysis, a method that laid the groundwork for modern genetics. Mendel's first law, the law of segregation, explains that alleles separate during gamete formation, so each gamete carries one version of a gene. A heterozygous cross typically yields a 3:1 phenotypic ratio and a 1.2.1 genotypic ratio, showing how recessive traits can skip a generation and reappear predictably. The law of independent assortment describes how genes on different chromosomes sort into gametes independently, which is why dihybrid crosses produce characteristic phenotypic ratios. By visualizing gamete formation and multiplying probabilities, we can predict combined trait frequencies, like the classic 9-3-3-1 pattern shown in the chart. Mendel's monohybrid cross between tall and dwarf plants revealed particulate inheritance. Crossing pure breeding parents produced uniform F1 offspring, and the F2 generation returned a roughly 3.1 tall to dwarf ratio. The observed data, with far more tall than dwarf plants, contradicted blending theory and supported the idea of discrete hereditary units. The test cross is a practical tool to distinguish a homozygous dominant from a heterozygote by crossing the unknown with a recessive individual. If all offspring are tall, the parent is likely homozygous dominant. If the offspring split about 1:1 tall and dwarf, the parent is heterozygous, revealing hidden genotypes with simple results. Sutton and Boveri provided the missing physical link by showing that chromosomes behave in meiosis like Mendel's abstract factors. Observations of homologous pairing and separation during meiosis, eye-matched segregation, and independent assortment, establishing the chromosome theory of inheritance and uniting genetics with cell biology. The Punnett square is a visual tool for predicting how parental alleles combine and what proportions of offspring genotypes and phenotypes to expect. By laying out possible gametes and their fusions, you can read off probabilities for single or multiple traits, making Mendelian logic and random assortment easy to apply. Mendelian patterns have clear clinical relevance. Recessive inheritance explains conditions like cystic fibrosis and certain metabolic disorders, while dominant inheritance underlies diseases, such as Huntington's. Using pedigrees and carrier analysis, genetic counselors can predict risks and inform patients, illustrating how basic laws translate into medical practice. In agriculture, Mendelian principles enable selective breeding and hybrid seed production. Predictable segregation helps combine disease resistance and high yield, while crossing distinct homozygous lines produces uniform F1 vigor. These techniques have been essential for crop improvement and remain a pillar of efforts to enhance global food security. Beyond Mendel, real-world genetics reveals exceptions that expanded the field. Linkage violates independent assortment. Sex-influenced traits depend on gender. Incomplete dominance produces intermediate phenotypes. Epistasis and lethal alleles alter expected ratios. Studying these exceptions led to deeper models and the discovery of broader genetic mechanisms. Mendel's framework introduced quantitative thinking to biology through rigorous data collection and statistical analysis, establishing discrete hereditary units and predictable transmission patterns. That analytical foundation paved the way for gene mapping, population studies, and modern tools like CRISPR, securing Mendel's role as a cornerstone of genetics research.